So today I want to focus on the recent work we did together with excellent group from S region. Now it's part of Leica system. The development of the deep learning methods for denoising and enhanced resolution over three-dimensional imaging volumes. For people who are familiar with the microscope imaging, the ideal imaging conditions we would expect to give us the high temporal and spatial resolution, and meanwhile with a very minimum light exposure. So in this case, the sample will be preserved very well during the imaging process. However, in reality, one has to sacrifice one condition or another to achieve this ideal imaging conditions. This is also known as frustration of pyramid, and basically it means no single parameters can be obtained without compromising the other factors. For example, you're either getting high spatial resolution and not the temporal resolution, vice versa. So for the past decades, there was a large effort that has been focused on the developing the computation methods as well as advanced imaging technologies to achieve this ideal imaging goal. The classical imaging degradation were attributed to the following categories. For example, extra noise, motion blur, and quantization, optical blur, and extra background. And one of the classical solutions to solve this problem for the microscope imaging is called a deconvolution. And the equation that I showed here in the slides that describes the typical procedures. So the degraded imaging G is equal to the convolution between the true imaging and the point spread function of the microscope. Then we could do the Fourier transform. We could convert this convolution function into a simple multiplication equation in the frequency domain. So in this case, we get the degraded imaging G equal to a simple multiplication between the desired imaging and the Fourier transform of the point spread function. So through the simple Richard-Lucet iterative deconvolution loop, we can effectively remove the noise from the degraded imaging. However, in order to get the deconvolution works great for the microscope images, you have to have a very good knowledge for the point spread function. On the one hand, the point spread function changes in different z depths when you're imaging the sample. On the other hand, when you acquire the image, if your signal is not bright enough, the signal is typically contaminated by the Gaussian and the Poisson noise. And those two factors together contributed significantly to get a very good deconvolution. So on the other hand, the artificial intelligence has played a more and more important role for the biomedical imaging process and imaging denoising. So machine learning is one category of the artificial intelligence, and deep learning is a small category of the machine learning. And basically, it can utilize a large amount of data to create to automating the creation of the software. And deep learning could really change every industry. So here is a typical flow chart of people who are using the convolutional neural network for the imaging denoising. What we typically do is that showed here is that we supply the convolutional neural network with noisy images, and the noisy image will predict restored images. And then through comparing the restored image and the ground truth, the network parameters will be updated to minimize the difference between these two images. Then we can repeat this process again and again until the training is finished. Once we obtain a trained neural network, we can then apply the neural network to the data set and evaluate how does a neural network works for the imaging denoising. So through a collaboration with excellent team led by Luciano from S Vision Technologies, we extend the existing residual channel attention networks RCAN into 3 dimensions, so we call the 3D RCAN. For the imaging denoising, and that's what the first one trying to solve. So this is the architecture of a 3D RCAN. Basically, what you can see here that the 3D RCAN composed of a few residue groups, and there is a non-skip connections between this to bypass these residue groups. And inside each residue groups, there were several residue channel attention blocks, and there was a short skip connections within the residue groups. So the design of these non-skip connections and the short skip connections was to bypass the low frequency learning and focus on learning the high frequency information, because most of the super resolution imaging was most relevant to the high frequency information. So, so the first thing in order to test this idea, the first thing we want to try was to see if we can use and train the 3D RCAN for the super resolution imaging denoising. So in this case, we turned into a microscope technology called the instant structural illumination microscope. For people who are not familiar with these technologies, you can actually go and check this was there was a publication related to this technology in Nature Methods. Basically, the combination of the multifocal excitation lens and the pinhole and emission microlens array, so enable us to get a resolution around 140 nanometer laterally and 350 nanometer axially, and we can achieve very fast imaging around 100 hertz. And then gamma middle will sum up all the images. So you can basically, this microscope allows us to do very fast super resolution images. So the image I showed in the next slides here, those are mammalian cells U2OS transfected with Tom20 GFP for mitochondrial staining. And in the first row, in the first column, you'll see here, those are raw data, and in both XY view and in XZ view. In the middle column, those are ground truths. Basically, those are same field of view, but excited with a much high excitation power. So you can see much better structures, and we can clearly see the resolution. Although the iSIM in principle should provide us 140 nanometer resolution in XY, but because of the noisy, and those resolution was buried in the noisy images. And in the third column, those are the 3D RCAN denoised images, and we can see that the 3D RCAN denoised image provide very clear improvement compared to the raw data set in both XY view and XZ view. Then we benchmark the performance of the 3D RCAN against another a few other state-of-the-art convolutional neural network. For example, one of the famous unit-based care, it's called content-aware image restoration. This paper was published in 2018 in Nature Methods. And also we compare the RCAN performance against SR resonant. It's a super resolution resonant, and the ESR again, it's an enhanced super resolution generative adversary network. So you can see actually all the 4 neural network provide very clear improvement compared to the raw data set. We then quantified their performance using the 2 metrics. One is a 3-dimensional structure similarity index. Another one is a 3-dimensional peak signal noise. And in the case of Tom20 here, we found that the RCAN provides a better network performance compared to the other network. The next question we want to ask is that, how does the RCAN denoising for the different cellular structures? Then we trained this 4 different convolutional neural network with different cellular structures ranging from the actin in the top row, and second row is endoplastic reticulum ER, and the third row is Golgi, next is lysosome and microtubule. And we also evaluated the network performance using these 2 metrics. We found that the RCAN works great for the variety of different cellular structures, and the performance of a 3D RCAN is competitive with other state-of-the-art convolutional neural network. Another important question for biologists is that so in the restored images, we actually a lot of times we want to quantify the fluorescence intensity because the fluorescence intensity also carry valuable information for biological questions. In order to answer these questions, we quantified basically different region of interest in the RCAN restored images versus the ground truth images. So in the image I showed here, you can see that the RCAN in the restored images, the fluorescence intensities match the intensity from the ground truth. In both TOM20, which label the mitochondrial outer membrane as well as a lysosome, and we also examined the line profile of different regions. So in the example I showed here, in both two cases, the RCAN fluorescence intensity from the RCAN image matches the ground truth images. So this results suggest that indeed we can utilize the counts information that from the restored images because those are important for some of the quantification fluorescence microscope. After we successfully trained the model in the fixed cells, we then apply the model in the fixed cells and to the live cell. So the example I showed here in the top row, those are live cell, and again, these are U2OS cells transfected with another mitochondrial marker. It's a matrix mitochondrial marker to so you can see the mitochondria diffusion dynamics in the live cells as a function of time. We are using very low excitation power because it was well known that the structural illumination microscope could provide us a better resolution, but the photobleaching is also an issue. And in contrast, the light sheet does not provide such high resolution, but the photobleaching and the photo damage is much less severe compared to the structural illumination microscope. So we found that a combination between the structural illumination microscope and the deep learning will enable us to operate the structural illumination microscope type instrument as light sheet microscope type instrument. The image was acquired like with 2,600 three-dimensional volumes, and each of the volumes contains 24 planes every 3 seconds. So this translates to, in a total, about 50,000 images in a window of 3 hours. And we can see that it's difficult to distinguish the individual mitochondria from the raw data, whereas this information is much more clear in the restored images. So we quantified we can segment the mitochondria from the restored images, whereas this task is difficult or impossible from the raw data set. The next application example, we then applied the 2 different models we trained in the fixed data set and into the live cell. In the left side of the images you see here, those are mammalian cells transfected with a lysosome as well as mitochondrial matrix protein. So the green color is mitochondrial matrix protein, whereas the red color is the lysosome. As you can clearly see from the raw data set, no useful information can be extracted. Whereas in the deep learning restored images, we can clearly track these 2 organelle interactions and dynamics. So we actually did quantify these 2 organelle interactions as a function of time. So we observed that, for example, in some of the cases, the lysosome was in the process to help the mitochondrial fission. We can see that the lysosome helped to create the bricks in the mitochondria, and the mitochondria was divided into multiple segments. Whereas in some other cases, we found that the lysosome will help to the mitochondria to fuse them together, and this is called a fusion. And in other cases, we found that there was simple contact between these 2 organelles, and then we can quantify like what's the fraction of these events against others. I want to emphasize that this quantification and track how does organelle movement in the live cell with this kind of resolution is difficult in the raw data set for such a long-term acquisition window. So another important question is that how much we can trust the deep learning restored images. So in the practical, in the classical methods, the people typically train independent convolutional neural network. For example, here we train 4 different independent RCAN convolutional neural network, and each give us two predictions. Then we can calculate the mean of those those 4 different predictions, as well as a standard deviation. And we can also calculate the ensemble discriminant. And however, in practically, we found those mean, standard deviation, and ensemble discriminant does not give us very rich for information to predict in the regions that actually the RCAN prediction in the regions does not match the ground truth. In contrast, we found that to calculate the signal to noise and the pixel basis give us a better predictions about the regions that will match or not match the ground truth images. So in the next image I showed here, we basically use the same raw data set and we just added a different noise level to the same raw data set, and each of them will generate 2 different RCAN predictions. And number 5 here in the slide showed here is a ground truth, and in the small image I showed in the region over here, corresponding to the yellow highlights. So as you can see that the RCAN predictions, the quality of the RCAN predictions decreases when the noise level increase. Perhaps this is not surprising, but if we use classic calculations, for example, the mean and standard deviation, as well as ensemble discriminant, we found that this is simply not correlated well with this kind of regions. And we found that estimating the signal to noise could be very useful to get an idea about the network uncertainty and give us a prediction confidence. So motivated by our denoising results, the next question we have in our mind was trying to ask whether we can use the 3D RCAN to enhance the resolution. So the idea was very simple. We supply the 3D RCAN model with pairs of low resolution and high resolution images, and then train the model to see if we can deblur the blurred images. And we also want to explore the limit at in what degree and to what extent the model will fail for the resolution enhancement. In order to answer these questions, we synthetically made some phantom sphere data, and we blurred this phantom sphere data 2 times, 3 times, and 4 times. As you see in the first column here, those are raw data set, but those are gradually blurred. And the second column, those are ground truth images. And the rest of the column are 4 different convolutional neural network images. We found that all the 4 convolutional neural network works great for the 2x and 3x blurred image. Whereas we see that although the RCAN provide the best deblur results compared to the other convolutional network, it's still like fair to recover some of the phantom particles in the 4x blurred data set. So here is a movie to show that the XZ view of the input 4x blurred raw data set, as well as the other convolutional neural network deblurred data set. And as I mentioned in the previous slides, the RCAN, although RCAN provide the best deblurred results with 4x blurring data set, we still can see some of the regions that the RCAN is failed to recover. So it looks like from this synthetic data set, the 4x blur is sort of the limit that we can use the RCAN for the resolution enhancement. So motivated by this statistic data set results, we then apply this 3D RCAN to the confocal images, and we want to ask if the 3D RCAN can learn the STED images from the confocal images or not. So we basically train the RCAN with pairs of confocal and STED images together. And in the example I showed here, those are two examples. One of the on the left side, those are mitochondria, and on the right side, those are nuclear pore complex. And you can see in the middle row, those are RCAN predicted STED-like images. Compared to the confocal input, the RCAN predictions provide very clear resolution enhancement in both XY view and XZ view. There's also some regions that the RCAN was fair to predict, and through quantification, we found that the RCAN provides 2.5x resolution enhancement compared to the confocal microscope images. And this is particularly attractive because the confocal microscope provides much higher signal noise, and also it's faster, but it provides much worse resolution compared to the standard images. So we can utilize this coarse imaging modality transfer learning and to get a STED-like images from the confocal images. So in the third example, we trained RCAN with pairs of different mitotic stage from the mammalian cells. And this nucleus was stained with a DNA staining dye, silicon-rhodamine, and you can, as you can see, the different mitotic stage in the image that's showed here. So this model was trained. After that, we applied this model to the live cell resonant confocal images. We found that we can nicely predict the STED-like images based on the resonant confocal images, and the chromosome and nucleus structures was poorly defined in the resonant confocal images. And in contrast, this resolution in the chromosome and the nucleus was much better resolved in the RCAN predicted images. So another important aspect I want to mention is that although these two different imaging modality was acquired on the same conventional instrument, there was still we found that the alignment between these two imaging modality was necessary to help further improve the imaging quality. And we did the experiments. We showed that the aligned images between the confocal and the STED images before training versus the unaligned confocal to the STED images. We found that affine registration between the confocal and the STED training data set helps to improve the prediction images. So after we saw that the RCAN was able to successfully to enhance the resolution from the confocal to STED images, we wonder if we can further increase the resolution we get from the iSIM images. So one of the ideas come to our mind was to use an expansion microscope. So instead of imaging the sample, we actually put the sample into the matrigel. For the people who are not familiar with expansion microscopes, the basic idea was that you embed your sample in the matrigel and then you physically expand your sample isotropically. This expansion factor was determined by the fetal by the gel, and then so in this case we can basically obtain a high resolution. This is the workflow to show that how we get the training data set for the iSIM to the expansion microscope. So the basic idea was we first grow the cells on a glass bottom counter dish, and then we image them in a no magnification lens. We then find the region of interest. We then cut those region of interest, and then make the expansion gel, and then we digest and expand the expansion gel. After that, we image this the gel in a new glass bottom dish, and then in the high magnification. So in this case, we can get post expansion images. So we then found that the alpha registration between those post expansion images and the pre expansion images is difficult. So we end up using the synthetic blurring and adding noise to the post expansion images to make the synthetic raw data. And we found that the synthetic raw data and the post expansion images could give us a very good training data set to supply to the RCAN model, so we can get expansion models. After that, we apply the two-step RCAN to the iSIM images. So we first apply the denoise model to remove the noise, and then we apply the expansion model to get the predicted expansion images. So in the example I showed here, those are two-step RCAN expanded images. On the left side, those are top 20 mitochondrial images, and the right side, those are mitochondria expanded predicted images. We can see that in the two-step RCAN images, it provides a much better resolution improvement in both XY and XZ view. We then further quantified how much improvement we can get in terms of resolution compared to the iSIM. So we found that we can get 1.9, almost 2x improvement naturally, and 3.6x improvement actually compared to the iSIM images. We then apply the expansion model. We trained on a fixed cell into one of the live cell data set. Shown here are the Junker T cell, Junker T cells expressing the microtubule fused with 3x GFP, and this Junker T cell immune cells are settles on the anti-CD3 coded activating cover slips to mimic the antigen presenting process. So you can see the microtubule are spread nicely on the cover slips. And in the two-step RCAN results, we can resolve this microtubule structures much better compared to the iSIM images. So in summary, I talked about the RCAN to enable the denoising, and its performance is competitive with other state-of-the-art convolutional neural network. And the RCAN course also enables us for the resolution enhancement. It provides 2 to 3.6-fold resolution improvement for the Confocal and iSIM dataset through the cross-modality transfer learning. We also talk about by combination of using the RCAN and the iSIM, we can operate the structural illumination microscope type instrument as a light sheet type instrument. And last, we talk about to using the signal to noise for evaluating and predict the network performance. And we have the code and the source data available from the GitHub. And for people who are interested to apply and test this 3D RCAN network, here is a link. Last, I want to thank for all the people who attended my talk, and also I want to thank for all the people who contributed significantly to this work. The people from our team AIM, and also Harry Schroff and lab members from Harris Lab, and the excellent team from S-Vision Technologies, and people from NHLBI, as well as people from University of Maryland. And thank you very much. I'm happy to answer any questions.
Well, Jiji, thank you so much. Really great to see the work presented by you. It has been, and it continues to be a pleasure to collaborate with you and Harry and the broader team. And of course, we look forward to continuing that and growing that. I do have a question, and it's maybe an invite for a comment from you, because there's a lot of approaches for denoising now published. Aftercare, there's a few more, and then there's this one. Where do you see things evolving to? And I'm specifically thinking about the characterization and for real biology experiments, the characterization of the limits of these networks is, I would say, at the core of the issue. So if they are well characterized, they may be used. They're not well characterized, they cannot be used, probably. But I'm wondering, what you think, and if you could share your thoughts on, from a biologist point of view, on this topic.
That's an excellent question. From my understanding, I think convolutional neural network will do a great job to answer some of the binary questions. For example, for detections, whereas the biologist is not care so much about the network limits. If you want to answer if the signal is on the plasma membrane or if the signal was on a specific organelle or not, and that I think is less of the concern of here. But for people who want to use and apply convolutional neural network for the super resolution imaging, those are the people who care about the structures and care about the dynamics. In these cases, I think people need to be very careful about applying this network. As we showed in the talk, like there was a classic quantification methods tend to fail in those predictions. And if you're using those network for the very poor signal to noise images, you need to be very careful and to interpret this data set. And for in the cases of you want to simply just track the movement of the organelle, and in those cases, I would say that maybe if there was some, even if the network generates some of the artifacts in the structures, you care less about those artifacts. And in this case, I think biologists will feel very comfortable to use this neural network. So it's really like a case by case and depending on the application. And further down the road, I think they need a better work to further push the limit and to see what was the limit of this convolutional neural network. And the work we did together here was a supervised work. And I think in some of the cases, people does not have a training data set. And in those cases, unsupervised convolutional neural network will play an important role. That's what my take on the application side.
Okay, very good. Thank you for sharing your thoughts here. I appreciate that. Let me see if there's other questions. I want to give a moment for people to ask. Oh, there's one question coming here. I don't know if you can see it, so I'll read it out loud. Have you tried using Fourier shell or ring correlation method to quantify resolution improvement by RCAN? Might it be helpful?
Yeah, so we actually use a published method called a decorrelation analysis, and it's sort of similar to what he mentioned here. Yeah, I didn't go to the details about the quantification, the resolution quantification in my talk here, but the method we use was decorrelation analysis, as published in Nature Methods a few years ago.